Bismillahirrahmanirrahim. So today we are going to study about the Shigella. Shigella. Although Shigella is divided into many species, but the two medically important species are Shigella soni, and the Shigella dysentery. So we will mainly focus on these two species. So starting with the main introductory point regarding the Shigella. So the first point regarding the Shigella is that it is a gram-negative straight bacteria. It has a straight shape and it belongs to a group which is known as Enterobacteriaceae, like the Escherichia coli, which we have already discussed, the Salmonella. It also belongs to a group known as Enterobacteriaceae. So thus it consists of all the features which are the features of Enterobacteriaceae, like it is a facultative anaerobe, it is oxidase negative, and it reduces nitrites to nitrates for the purpose of energy production. And the fourth one, it is a glucose fermenter. It is a glucose fermenter, but it is a non-lactose fermenter, except the Shigella soni, which is a delayed lactose fermenter. It will require very long time for the fermentation of lactose. So it is a delayed lactose fermenter. The third important point regarding the Shigella that they are all citrate negative. And they do not use the citrate for the purpose of their energy. They are citrate negative like the Escherichia coli. The fourth important point is that they are non-motile, and they do not produce any H2S. Unlike the Salmonella, which produce H2S, they do not produce any H2S. So these were some introductory points regarding the Shigella. Now let's come toward the antigens which are present in the Shigella, the important antigens. It consists of three antigens, like the Escherichia coli and the Salmonella. It consists of three antigens. The first one is for cell wall antigen, and on the basis of this, the Shigella is divided into three subgroups: the A subgroups, the B subgroups, and the C subgroups. It also consists of the K or capsular antigen, and also consists of flagellar antigen, but they are not important like the cell wall antigen, on the basis of which they are divided into subgroups like the A, B, C. Now let's talk about the epidemiology. So the Shigella, they only cause human intestinal tract disease, and they are mainly transmitted through the 4 F's, like the feces, food, fingers, and the flies, and only cause, as I have already said, they only cause the human gastrointestinal tract diseases. There is no carrier state. Now let's come toward the diseases caused by the Shigella genus. So the first disease caused by the Shigella, and the most important one, is the Bacillary dysentery, which is also known as enterocolitis. So what is the pathogenesis behind the Bacillary dysentery? How the Shigella caused the Bacillary dysentery? So it is mainly through two mechanisms: by the invasion, which is the main one, and the least common one is by production of Shiga-like toxin. First, talking about the invasion, so this Shigella, they mainly invades the mucosa of the distal ileum distal ileum, and colon. And after invasion, there they produce the inflammation, and cause the proliferations and the infiltrations of the neutrophils, and thus cause the ulceration, and thus it ends in the bacillary dysentery, which are mainly characterized by the symptoms like the fever, abdominal pain, diarrhea. And this diarrhea, it is initially watery, and later it becomes the bloody, and this is mainly due to the inflammation. The watery diarrhea is produced because of this Shiga-like toxin, which do what? It cause watery-like secretion from the intestine. And this bacillary dysentery may be mild or severe. This mild case is mostly occur in the young peoples, and it is mainly caused by the Shigella soni. And the severe one mainly occur in the old people and the children, and it is mainly caused by the Shigella dysentery. And this Shiga-like toxin is only produced by the Shigella dysentery type 1. It is only produced by the Shigella dysentery type 1. And this enterocolitis also has an incubation period of about 1 to 4 days. Now the second disease caused by the Shigella is the hemolytic uremic syndrome. As we have already discussed, and this hemolytic uremic syndrome is characterized by the features like hemolytic anemia, the acute renal failure, and the thrombocytopenia. I have already discussed the hemolytic uremic syndrome in the diseases caused by this Escherichia coli. So in order to understand the pathogenesis and the clinical findings of hemolytic uremic syndrome and the treatment of hemolytic uremic syndrome, only watch its videos. So it is the second disease caused by the hemolytic uremic syndrome. Now let's talk about the lab diagnosis of the Shigella. Lab diagnosis. So for the lab diagnosis, the first one is what? Is the microscopy. In the microscope, we will see for the gram-negative straight, gram-negative rods. The second one we will see for the pus cells and for the RBC in case of bacillary dysentery. We can also do cultures, and the important one in the cultures is the McConkey agar. And on McConkey agar we will see what? Colorless colonies. Except in case of Shigella soni, which is a delayed non-lactose fermenter. So after some time there will be pale pink colonies in case of Shigella soni, which is a delayed lactose fermenter. We can also do TSI agar, and TSI agar there is a slant and butt. So on slant it will be alkaline. I.e. it will be red, and the butt will be acidic, i.e. it will be yellow, and there will be no gas and H2S production. There will be no gas, i.e. there will be no bubbles, and H2S, i.e. there will be no blackish color in the butt, and the slant will be alkaline red, and the butt will be acidic, i.e. it will be what? It will be yellow. We can also do semen citrate test. It is mostly negative because they do not use citrate for the purpose of their energy production. And for the serological purpose, we do the latex agglutination test. Now let's come toward the treatment. So in case of mild cases, we only do what? Fluid and electrolyte replacement, and there is no antibiotic. No antibiotics are given. In case of severe cases, there is fluid and electrolyte replacement, and the drug of choice is the fluoroquinolones. Fluoroquinolones. And in case of resistance to fluoroquinolone, what we do? We do trimethoprim, sulfamethoxazole. Sulfamethoxazole. The drug of choice is fluoroquinolones. In case of resistance to fluoroquinolones, we give trimethoprim, sulfamethoxazole. And the important point is, note that antiperistaltic drugs are contraindicated. Antiperistaltic drugs are contraindicated in case of the bacillary dysentery because it will prolong the fever and the diarrhea, and there will be excretion of organisms in the stools, and it can serve as a source of Shigella bacillary dysentery transmission. So that's all for today. Thank you.